Okay, we're back. So let's review a little bit. What did we talk about yesterday? So we talked about the upper digestive tract. So where were we? We talked about the mouth. So this is where digestion starts. You've got salivary amylase. That's the enzyme that's released here that starts to break down starches. So those are carbohydrates. So you've got chemical digestion and you've got mechanical digestion with your tongue and your teeth grinding food down, breaking it down, turns the food into a bolus. There's also water in saliva here. So you've got salivary amylase and water. What's that water for? Remember what that water's for. You swallow it down. The pharynx here is where the trachea, where you breathe, is connected, and the esophagus is connected. You have the glottis here, that's the opening to the trachea, and the epiglottis, this is this flap of skin. So when you swallow, the epiglottis folds over the glottis and down the food goes. The food moves down the esophagus via peristalsis, which means we can swallow upside down. And so the bolus moves down the esophagus, and it ends up stopped here at the cardiac sphincter. Is that right? There we go, like that. So it ends up here at the cardiac sphincter, and then it enters the stomach, and the stomach, it's got 3 layers of muscles, not just 2 like the esophagus, so it could squeeze and then it can churn the food. So there's mechanical digestion going on here. Also, the stretching of the stomach releases it stimulates G cells to release gastrin, which then stretch receptors make the endocrine G cells release gastrin, which then stimulates exocrine glands in here to release gastric juice. And gastric juice is a combination of acid and water and pepsinogen. I don't think I talked about pepsinogen, but pepsinogen is a precursor to pepsin, which is a peptidase that breaks those peptide bonds in proteins. It turns them into amino acid, makes larger proteins into smaller peptide chains. So pepsinogen is a precursor. Why it's a precursor? Because it's really dangerous. It eats protein. So if it was in your cells as already pepsin, it would start eating your cells and the DNA inside the proteins inside your cells. So it's released as a precursor, and then the environment in here triggers it to become pepsin. So the acidic environment turns it into pepsin. So that's the stomach. So here the stuff is being churned. Another function of the stomach is part of your immune system. We shouldn't forget that, is because it's acidic in here. You've got your trachea. So when you breathe in bad things, dust, anything, there's little cilia hairs all along your trachea that capture that stuff, and those cilia, along with mucus, push the bad stuff up and into the epiglottis. No, not the epiglottis, into the esophagus, and you swallow the bad stuff down, and it goes into the stomach acid and gets dissolved. Gets dissolved by the acid. So that's part of your immune system. If you damage these cilia, you're damaging a very important part of your immune system. That's why smoking is so bad, because the tar from the smoke will cover the cilia and stop them from working. Smoking anything, even smoking marijuana, will damage the cilia, the heat, the smoke, and you will be more prone to coughing fits and diseases of the respiratory system. Okay, so we're in the stomach. Here's the pyloric sphincter. That's the sphincter at the bottom. And so it is closed and food is being stored. Then you need some more energy. So actually food is stored inside your stomach. So you need some more energy. Your food gets stored or it's just the time for it to move through. so the pyloric sphincter opens, and what's called now acid chyme, because it's been mixed with, so we had the bolus, now the food has been turned into chyme, and it's called acid chyme, and it enters here into the upper intestine or the small intestine. And so we're going to move actually the small intestine out of the way. This model, the intestines are not the greatest, but you can see that was just a curve of the large intestine. But so here, this is the duodenum over here. This is the start of the small intestine, and it connects here, and then the small intestine wraps around. So that's the duodenum is the first section of the small intestine. Okay, so what happens in the duodenum? A lot of stuff happens. Duodenum, important place. So here's the duodenum. So number one, now you've got acid chyme entering the duodenum. So the food that was in here, we had the food bolus. It's now protein digestion has happened here, and the food's been broken down more. Also, the acid has broken stuff down. So now it's called acid chyme. So acid chyme enters the duodenum, and there are chemoreceptors, so receptors that respond to chemistry, basically, in the duodenum. Let's see, I can move some of this intestine out. So there's the duodenum. Okay, there it is. Can we see here? Let me just poke around in here. Oh, that's fine. Where's the liver? There's the liver. Okay, so we got the duodenum. So there's chemoreceptors. So chemoreceptors respond to the drop in pH, and they respond to just the molecules entering, like the food entering. And what happens here is that those chemoreceptors then stimulate two hormones to be released, and that they're released from endocrine glands. So they're released into the blood, and that is CCK plus secretin. And CCK does two things. So CCK goes into the blood, and it targets a part of the liver, actually, the gall bladder, this little thing. And the gall bladder stores something called bile. And bile is made of, one of the things that that happens in the liver is red blood cells are broken, worn out red blood cells go to die here. And there's bilirubin is what's left over from the red blood cells. There's two billies. Anyways, they make your poo green. They actually look green. They're like the proteins of the red blood cells. Anyways, this stores bile. They get turned into bile. Bile is a combination of stuff. And bile, when CCK, so food has come in, those chemoreceptors have detected food has come in, the acid has dropped, so bile is released down this duct, the bile duct, and it's released into the duodenum. And what bile does is it's like soap in a way. What it does is it breaks up fatty molecules, but it doesn't do it chemically, it does it mechanically. This is called emulsification of fats. So it emulsifies fats. So fats, this is, we're mostly made of water. Remember, water and oils don't mix. So what happens is the emulsification is that big chunks of fat get broken down mechanically. The emulsifiers break them down into smaller chunks so that they're a manageable size then for the enzymes to then chemically break down the fats, right? So those triglycerides. So you have glycerol and those three fatty acids, they can break it down then. But the fats don't mix well with water, so they have to be first mechanically broken down. Okay, so that's the first. So you got bile duct spilling out bile, gross green bile. And then also you've got, so CCK, secretin, those were the hormones released by the from the duodenum there. They're endocrine glands, so they've gone through the blood. They also target an organ here called the pancreas, which you can't really see very well. There's no... Anyway, so the pancreas, what it does is, so secretin and CCK trigger the pancreas to release something called pancreatic juice. And that's, remember we had gastric juice? Just adjust I'm squashing here. So we had gastric juice. So this is pancreatic juice. And pancreatic juice is just a combination of a whole bunch of stuff. You got water again to do hydrolysis. You need water for hydrolysis. And you've got a whole bunch of enzymes. You've got lipases. So those are going to be the enzymes that are breaking up those fats, breaking up lipids, lipase. You got nucleases. So those are going to be breaking down nucleotides. They're going to be breaking down nucleic acids. So they're breaking down DNA into nucleotides and the component parts of nucleotides. You've got an enzyme called trypsin. And trypsin breaks down peptides. So just like pepsin was doing it in the stomach, trypsin does it in the duodenum. And you've got pancreatic amylase. So remember, in the mouth, we had salivary amylase for breaking down carbohydrates, for breaking down the starches. So now we've got pancreatic amylase for breaking down starches, continuing that process here in the duodenum. You also get a whole bunch of bicarbonate. So HCO3. The reason for that is a buffer, a pH buffer. That acid chyme has come in here, and it is very acidic. All these enzymes don't work in acid. They need a more basic. So actually, all that bicarbonate gets dumped in. It soaks up. Remember, we learned at the beginning that when something's acidic, it means it's got a lot of hydrogen ions. So bicarbonate soaks up HCO3 minus. It soaks up those loose H's, and the pH rises in the duodenum to around 8. It's a pH of 8. So it's not neutral, it's actually basic. So your food contents go from like a pH of 2 something to a pH of 8. And now the food is no longer called acidic chyme. It's called just chyme now. We lose the acid chyme bit. And so now the pancreatic juice, you've got those lipases breaking down fats. You've got to their monomers, right? You've got nucleases breaking down nucleic acids. You've got trypsin breaking down, you got trypsin breaking down peptides, so proteins, and amylase breaking down carbohydrates. That's happening in here. So you need water and stuff to do that, okay? So that's happening in the duodenum, and as it travels down the duodenum, all that's going on, and that continues on throughout the small intestine. But most of the is there anything else I'm forgetting? Pancreas. The pancreas also, so there's a whole bunch of stuff that the pancreas does. Pancreas also secretes insulin and glucagon, and these go to the liver. And basically, you know how we store sugars and glycogen, so those sugars, so the glucagon and insulin control how much of that is being released into this. So we don't have enough. If your blood sugar is low, then it will release insulin, which breaks this down. It helps sugar go into your cells, so you can get more energy. But anyways, continuing back on here, so most of the first section of the small intestine is where most of the digestion actually happens, that chemical digestion. And then the rest of the way what's happening is absorption. So the small intestine has something called villi, so it's lined with little hairs. And what do these do? What I say is always the answer is surface area. They increase surface area. So you've got these villi and microvilli, which are even smaller little protrusions. They're not really hairs. They're like protrusions of the lining. It's like folded up skin. These little things sticking up all over, tons of them, all over the inside of the small intestine. And so they're increasing surface area because that way, and there's blood vessels that run inside these villi, all the blood vessels, and so the cells along the villi, those cells across their membrane, they've got proteins, transverse proteins that recognize the stuff that the body wants and absorb, so they suck in the carbohydrates, the amino acids, the nucleotides, all that stuff is going in, fatty acids, all of those things. And this is, remember, a semi-permeable membrane. So things that you don't want, you're not going to take those in. So I know there's a whole industry about talking about toxins in your food and stuff like that. How do they get into your, because we're still actually not in the body. It seems like we're in the body, but remember, we've just been traveling down a tube that is connected to the outside world. Only now are things entering the body. They're being they're moving across the membrane of the small intestine, that inner lining of the small intestine and those villi, and now they enter the blood. They also enter the lacteals, which is part of the lymphatic system. So the fats enter. There's inside. If you imagine this is one villi, inside is a tube called a lacteal. This is going to be important when we study the lymphatic system. And so the fats enter here. They're absorbed in here. So there's the toxins from your food or whatever, you just poop them out. They don't make it in. So, the value of a cleanse, I'm suspicious of it. Trypsin, there is a precursor to trypsin, which the name of it escapes me right now. Trypsinogen. So there we go. So the protease trypsin, it comes out as trypsinogen, and actually what happens is here it's not activated by the environment. This is the important part. It's activated by something called Enterokinase, and so that's an enzyme that's in the duodenum that's also released by the pancreas, and it snips a portion. It'll be like the active site of trypsinogen is covered up, and Enterokinase snips whatever is covering the active site, so that trypsinogen can become trypsin and start digesting peptides. So you can see its level of control. And if you have mutations, if you have problems with enterokinase or trypsin, it'd be hard for you to digest protein. So this is going to be a long video. It's already, I have to edit it. So the rest of the small intestine, all those nutrients are being digested, and by the end it gets to the ileolacteal valve, another sphincter, to enter the large intestine. So the large intestine, there it is connected. All large intestine is here. It all curls around. And what happens in the large intestine, so you've already absorbed all the nutrients, but there's a whole bunch of water. You've been pouring water into here for hydrolysis. So in order for hydrolysis to happen, remember that chemical reaction of breaking down polymers into monomers. You want to take that water back. So the large intestine, one of its main roles is just absorbing water, just taking water back out of that chyme and starting to turn the chyme into feces. There's also bacteria in here, and these bacteria, they are our friends. They're our friends, and they help break down some things that we can't break down. And basically, they break that down, and then they release amino acids and vitamins that we absorb and use. They're anaerobic, these bacteria, so they don't need oxygen, and they are symbionts. So we have a symbiotic relationship with them. What else is happening? So bacteria, basically nutrients, amino acids, and minerals, and water. That's the big thing about the lower intestine. And then, so that stuff, it's getting dried out. The bacteria are changing it. Then we're giving it some stink, because the bacteria, they'll get caught up and they output gases that give the feces some stink, and it gets stored in the rectum. And then when you're ready to poo, you poo it out. Don't poo where you eat. So there'll be lots of bacteria in that poo, and it'll be yucky. And that's it. That's digestion. That's all there is. So things to keep in mind, things that I haven't talked about here. I haven't talked about enough about the liver, so have a look at stuff about the liver, but also just track each biomolecule. Good idea to track each biomolecule. So what happens? Carbohydrates, they get broken down in here by salivary amylase, and then here pancreatic amylase breaks down more carbohydrates. So they seem to be pretty easy to break down, but there's two places where they get broken down. So, and they get absorbed. Fatty acids, so lipids, fats. They don't get broken down here. They don't get broken down here. They get broken down here in the duodenum. You need bile. You need bile from the Gall Bladder, and you need lipases from the pancreas. And the bile mechanically breaks down the fats, and the lipases chemically break down the fats. What else are we talking about? Proteins. Proteins get broken down in the stomach. Pepsin. And so know what's the story of pepsin and Trypsinogen. So those get turned into smaller peptide chains, and then there's trypsin in the duodenum, in the small intestine. Trypsin breaks those proteins down even more. So and then nucleotides, which we don't eat a lot. That's not a major component. We can get energy from all of those things. We can even get energy from those nucleases. And the other thing is to think about, where do those things go? What are they used for? So where do the carbohydrates go that those amylases break down, right? They go into the blood. First they get absorbed in the villi, into the blood, in the small intestines. And then what happens to them? So some of them, those carbohydrates, go to get stored. It's glycogen, right? Put it for storage. But a lot of them are going to be used for energy, for ATP. The fats, again, they're going to go for storage, and they're going to be used also, those pieces, for making more cell membranes, and then for storage as fats as well. And they can be also metabolized for energy when we need to, when we're really starving. The nucleases, those nucleotides, where do they go? Well, they go to make new nucleotides, to make RNA, to make DNA, all those things that we need. The proteins, what do the proteins get broken down into, right? I said peptide chains, and then what do the peptide chains get broken down into, right? Amino acids. You knew that. And where do those amino acids go? Well, they go to make more proteins and more enzymes to run our body. So we're getting to all those things that I just said. I was trying to answer that question I asked yesterday, is why do we eat? Hopefully I haven't missed anything, but I have. So have a look at the notes. I'm sure I have. Have a look through the notes. Know the story of each biomolecule, how it goes through the system. So read through the book, answer those concept checkup questions. That was the lower digestive tract. We should talk about some of these. Pancreas. So, and there's that nice table here lined with Villi. So have a look through that. Those are really good. And the liver. I haven't talked enough about the liver. And that basically covers digestion. So stay tuned for what we're going to do next.